Welcome to Sarkia channel. Now, let's talk about Leishmania tropica. Leishmania tropica is a species of a parasitic protozoa that belonging to the genus of Leishmania, which causes a disease that known as cutaneous leishmaniasis. It means cutaneous is relating of the skin or in the human skin. There are three classes of leishmaniasis. First is the visceral, second is the cutaneous, and the third is the subcutaneous. Since our topic for today is Leishmania tropica, so it is belong now to the cutaneous, which means it is related to the skin. This disease is transmitted to humans through the bite of infected sandflies. Distributions of Leishmania tropica. So here in the map, as you can see, the color purple, it is the primary areas, which are the regions mostly that have Leishmania tropica, which are the Middle East, Central Asia, and the parts of the Africa. So what is the habitat of Leishmania tropica? Leishmania tropica is a cutaneous, which means in the skin. So it means that the habitat of the Leishmania tropica is in the skin. So the amastigote forms occurs in the reticuloendothelial cells of the skin, whereas the permastigotes form are seen in sandfly vector. So Leishmania tropica is they are mostly staying of the human skin. Morphology. So Leishmania tropica is similar to the Leishmania dunavi, which is in the class of visceral. The protozoa parasite exists in two forms, which is the amastigote and permastigote. So here in the picture, the long flagella is the permastigote, while the egg shape has a short flagella, which is the amastigote. So what are their differences? When we say a mastigote, it is a flagellar stage, while the permastigote is a flagellar stage. And when we say a mastigote, it occurs in the vertebrate host, while permastigote occurs in the sandfly. And a mastigote is a round or oval shape, while the permastigote is a spindle shape. So a mastigote nucleus relatively larger and situated centrally, while the permastigote is smaller and situated in the middle of the cell or along the side of the cell wall. Now the life cycle. So first is in the sandfly stages. So the sandfly takes a blood meal which inject now the permastigote in the human skin, and then the permastigote are phagocytized by a macrophages or other types of monocular phagocytic cells. And then the permastigote now will transform into amastigotes. So the amastigotes will multiply in a cell of various tissues and infect other cells. And then now the sandflies will take another blood meal and then injects the macrophages that is infected with a mastigote. And then after that, it will ingest of a parasitized cells. Now the amastigote transform into permastigote again, then the stage in the gut. And then lastly, it will divide in the gut and migrate to proboscis. Next is the pathogenesis. So this is the Leishmania pathogenesis. So as you can see in the picture, the sandfly first will bite, and then the amastigotes proliferate, and then it will taken up with a macrophagic cells and endothelial cells. And then after that, it lyses of amastigotes by macrophages. Then the parasite-induced damage could lead to granulomatous lesions and pyogenic infections, and now become ulcers. Next, what is the clinical features of Leishmania tropica? So if you have Leishmania tropica, it could lead in infections, irregular boost of fever, weight loss, and could lead also an enlargement of spleen and liver. And what is the mode of transmission of Leishmania tropica? A direct contact, man to man or the animal to man, and also a auto-inoculation. Next is the immunity. So what lymphocytes need to boost to fight now the Leishmania tropica? So the lymphocyte is the T lymphocyte or called T cells. So it is essential for coordinating now the immune response against now the Leishmania tropica. It can differentiate in different subsets such as CD4 plus T, the helper cells, and the CD8 plus cystotoxic T cells, and each playing distinct roles in immunity. Next is a diagnosis. So this is now to know if you have the parasite Leishmania tropica. First is the microscopy. This is now to smear from agerated nodules or edges of the source, stained by Leishmania stain, and a mastigote inside microscope, and then biopsy, culture, skin test, and serology. Next, how to treat Leishmania tropica. So, an anti-moni-resistant cutaneous is treated with pentadamine, and a topic treatment with the 10% charcoal is a sulfuric acid or a liquid nitrogen. Prophylaxis, control protective clothing and repellents, and also the eliminations of reservoir. And lastly is how to prevent now the Leishmania tropica. First is avoiding sandfly bites, so wearing a long sleeves and long pants, using an insect repellent, and during the peak or sandfly activity, typically down and dust, just staying indoors. Next is the vector control by spraying insecticides or environmental modification can help reduce transmission. Public educations, because this is now about the risk of Leishmania and prevent measures that can help reduce transmission. Also protective clothing, as I said earlier, by wearing clothing that covers the skin, especially during the outdoor activities in endemic regions that can help to prevent Leishmania tropica.